I'm David Christianson. I'm the Roy and Diana Vagelos Professor in Chemistry and Chemical Biology at the University of Pennsylvania in Philadelphia. I've been here for 37 years, quite some time. So my research at Penn has been 37 years. Prior to that, I was a graduate student and undergraduate student at Harvard, and I started doing research as a junior in the college at Harvard and stayed there for graduate school doing research. So altogether, many years of research.
That's very impressive. And then, how did you get interested in research in the first place?
It's chemistry and being interested in chemistry and learning new things. Well, what is research but learning new things? So I first learned about chemistry from my first high school chemistry teacher, in fact, but I was a second grader at the time. And so that warrants an explanation. My father was a music teacher at the high school. And our family friends were the chemistry teacher. After school, in elementary school, I'd walk over to the high school and wait for my father to doing chemistry experiments as a little kid. So it stuck. And as the years went on, I just learned more and more about chemistry and learned that research is a big part of what chemists do. Thought it was really fun and what I've been doing for many years. And in fact, it's so much fun. I don't feel as if I've really ever worked a day in my life. I'm just doing what is fun.
Yeah, that's so cool. And then, in terms understandable to a high schooler, could you give me like a quick overview of your research?
I'm interested in the structure and function of proteins, which are biological molecules. Most of them are pretty big. And in particular, I'm interested in enzymes. Enzymes are proteins that catalyze chemical reactions. That is, they make the chemical reactions go faster by 10 to the 10th, 10 to the 14th, even 10 to the 20th times faster than the uncatalyzed rates. So, how do enzymes do this? Well, we do experiments to probe the contributions to catalysis, which involves measurements, catalysis in the laboratory, studying reactions in the presence and absence of the enzyme, and also determining the three-dimensional structures of the enzymes. And we use either X-ray crystallography or cryo-electron microscopy to determine enzyme structures in my group and combine the structure with the functional data, we can actually see the enzyme, and then we can understand the functional data as we look at the structure and say, this part of the protein is responsible for general base catalysis, for example. It's just a special subset of catalysis. So we combine the functional measurements with the structure. So this is why I say I'm interested in protein structure and function, or also known as protein structure function relationships.
Yeah, that's so cool. What were some of your most significant findings or publications?
Oh gosh, over the years, there have been several. We determined the first structures of enzymes called terpene synthases. These are enzymes responsible for the generation of more than 100,000 natural products, some of which are important as biofuels, some of which are important as fragrances, flavors, food additives, for example. Others are important as pharmaceuticals. The anti-cancer drug Taxol is a terpene natural product that is used for cancer chemotherapy. And artemisinin is an anti-malarial drug discovered by Yu Tu, a Chinese scientist who was quite famous. And she won the Nobel Prize for her discovery of artemisinin as an anti-malarial agent. So we study the enzymes called terpene cyclases that generate the carbon skeletons of these fascinating natural products. And as I said, once we see the structure, we can understand how they work with regard to the catalyzing chemical reactions. So there's one example of certainly a big discovery that has led to many, many years of research in my lab and others. Another is solved the first structure of an enzyme in the arginase deacetylase family. And that was in 1996. And that set the stage for the study of drug design. Arginase can be a drug target for immune modulation. And not only that, the deacetylases themselves are targets for cancer chemotherapy, often called histone deacetylases. It can be targets for cancer chemotherapy and are being studied as targets for neurodegenerative diseases like Alzheimer's disease. So, with the three-dimensional structure of the protein in hand, we can look at the active site where all the chemistry takes place in an enzyme, and we can figure out how to design a molecule that will fit into that active site, just as if a key fits into a lock. So, the enzyme is blocked if the enzyme activity is bad for your health. So that's how a drug comes about, especially when it involves an enzyme inhibitor. And I think a significant percentage of drugs on the market are enzyme inhibitors. I mean, at least 20%, it might be more. So this is what we use our structures for, not only understanding how the enzymes catalyze reactions, but if they're medically important as targets for human disease, then we design molecules to block them, to try to achieve a healthy state for a human who might take an enzyme targeted drug.
Yeah, that's so interesting.
We've all heard of COVID-19. The Paxlovid contains an enzyme inhibitor that blocks one of the viral enzymes so that the virus cannot replicate. And Paxlovid actually has another molecule in it, so it's two different drugs kind of combined together. And the second drug is to block a liver enzyme that would metabolize, that would break down the enzyme inhibitor in Paxlovid, the antiviral compound. So in that case, you've got a double strategy. You block the virus from replicating, and to sustain the concentrations of active drug in your body, you block the liver enzyme temporarily that would degrade, that would metabolize that drug molecule. So it's quite a fascinating application and an important application of enzyme inhibitors that has certainly saved lives in the past 5 years that we've been dealing with the COVID-19.
Yeah, that's so amazing. And then what were some of the biggest challenges that you faced in research and how did you overcome them?
Well, as a protein crystallographer, the biggest challenge is getting crystals because we use a technique, we use X-rays. We take an X-ray beam, aim it at a crystal, and the crystal, because of the molecular order in the crystal, it diffracts, it scatters the X-rays in a very well-defined way. And from the scattering pattern, we can determine the molecular structure and actually see the molecule that we're working with in that crystal. The biggest roadblock, though, is getting good crystals when you're a crystallographer. And that really has been it, for these very many years. It's always been getting good crystals of your protein target. How do you get around that? How do you circumvent that problem? It's just trial and error. It's more art than science, growing crystals. And sometimes we try really crazy things. So, for example, we didn't discover this, another lab did, but taking a cat whisker and just kind of drawing it across a drop containing the protein solution, it will help nucleate crystals in some cases. So really some crazy ideas that actually work for growing protein crystals, but they're very important for protein crystallography.
Yeah, that's so cool. I just feel like that's like a really creative method.
Yeah, and it really takes out of the box thinking. Who would think of taking a cat whisker and adding it to your experiment? Yeah, this is a pretty crazy thing to do, but it worked.
And then what do you enjoy most about your research?
It's not only discovering new things, but also training and mentoring students in my laboratory, both undergraduate students and graduate students and postdoctoral students. For example, I have an undergraduate student in my lab. He just published, well, it's her second paper, but it's her first author paper in Proceedings of the National Academy of Sciences. She started with a gene sequence. She expressed a protein and purified it. She crystallized it and determined its structure. And it's a very important enzyme. It has two different activities all rolled into one protein structure. I'm quite proud of Kristen Osika is her name. I'm quite proud of her accomplishments. Taking a project, it's really a PhD level project from start to finish. And seeing students at all levels, undergraduate, graduate, postdoctoral levels, come into the lab, make these sorts of discoveries, and advance their own learning and skills as part of the research, I find that very gratifying.
That's so cool. And then which person or mentor in your field has made the biggest impact on you?
I'd have to say my PhD advisor, the late William Lipscomb at Harvard University. He took me into his lab as an undergraduate and he kept me on as a graduate student. I learned many things from him. and still draw on some of those lessons that I learned from him to this day.
And then were there any pivotal moments in your research career that made you change topics or want to pursue a certain topic? Or kind of like what led you to look at protein specifically?
Well, I was always interested in proteins from the first biochemistry course I took in college. We learned a lot about enzymes and proteins, of course, there. But even in high school biology class, we learned about proteins and protein structure. The different levels of protein structure, primary structure, secondary structure, tertiary structure, quaternary structure. And it was quite fascinating. And I suppose that when we're interested in structure, it didn't have Legos when I was a kid. They had things that were like Legos, though. And I always liked assembling, the blocks that assembled in very specific manner. So into three-dimensional structures. And I suppose maybe before I even learned what a protein was, I was very interested in how things were assembled, how little bits assembled into bigger things. And that essentially is what a protein is, a bunch of amino acids connected to each other in what we call a polypeptide chain. So, where is the moment that I became interested in enzyme catalysis? I really can't point to one thing, but just a sequence of experiences that just built on each other as life progressed and just led me into the field of protein science and especially enzyme structure and function. In terms of doing something new or doing something different, I suppose, there are different enzyme systems that we have studied. And what always attracts my attention is unusual or very interesting chemistry. So it's always the chemical reaction that attracts me to a particular enzyme, but it's always enzymes. I've never really worked with any non-enzyme proteins, but in terms of the reactions that they catalyze, we've covered a lot of different types of chemistry with the enzymes that we've studied. And it's really when it comes time to picking a new system or being interested in a new system, it's purely curiosity that drives that. As I said, you have a very fascinating or unusual chemical reaction. How does the enzyme catalyze that? That's how it begins with that very simple question.
Yeah, okay. And then where do you see your research field expanding to? So, like, what are some interesting topics that can be further explored within the next 5 to 10 years?
Oh, 5 to 10 years, gosh. Protein engineering, designer proteins that will catalyze reactions that are new to nature. One can use directed evolution for that. Professor Francis Arnold at Caltech won the Nobel Prize a couple of years ago for the discovery of directed evolution. Now, how do you direct new functions into enzymes? Well, you can do it by directed evolution. But you can also do it by structure-based design, rational design, looking at a structure and saying, if I make an amino acid substitution here, here, and here, I'll introduce a new activity into that active site. So there's two different approaches to designing new catalysts. So I think that's in the next 5 to 10 years, just building on the design of new and novel catalysts to catalyze reactions for which there are no known enzymes, naturally occurring enzymes to catalyze. I think that's one of the great futures in biotechnology for the study of enzymes. In terms of the techniques of structure determination, cryo-electron microscopy has become very useful, especially in the past 10 years, for determining very high resolution structures of proteins. By high resolution, I mean you can see how atoms are connected to each other to form the molecule. Cryo-EM microscopy, it's usually not as good as X-ray crystallography in terms of the resolution of the image, but I think cryo-EM, it keeps on improving, that in the next 10 years, I think it will rival consistently with high-resolution structures with good protein samples. But even beyond that, might have heard of AlphaFold, using AI to predict protein structures just from the amino acid sequence. And the folks that discovered AlphaFold just won the Nobel Prize as well. I think AlphaFold and computational approaches will play an increasingly large role in understanding protein structure function relationships and the de novo design of enzyme catalysts. I mean, David Baker at the University of Washington also won the Nobel Prize for de novo design of this nature. So I think just the sky is the limit in terms of where science will go. But in terms of chemistry, I think the next 5 to 10 years, we're just going to see a lot more with the design of new to nature catalysts, just using enzymes as scaffolds to catalyze very unusual chemical reactions that were never encountered in nature.
Yeah, that's so cool. I actually like just started a class like learning about AlphaFold. These are questions more geared towards high schoolers. So, how did you find your passion and how would you suggest a high schooler try to find their own passion in research?
I was lucky that I had good teachers that helped, that recognized the passion and helped fuel that passion. If you're interested in science, I mean, there are opportunities for doing research, even research in your garage at home. So when I was in high school, thinking back to my junior year, the summer of my junior year, remember it was a teacher who suggested that I do a project for science talent search, a statewide and a national science symposium. So I did a research project because at the time, this was 78, 79, there was an energy crisis at the time. And what does that mean, energy crisis? Middle East had sort of cut off the shipments of oil to the United States, so we didn't have as much oil products like gasoline. So there are lines at gas stations to fill up with gasoline. They had if your license plate ended with an even number or an odd number, that determined which day you could fill up your tank. I mean, it's really serious. We've seen nothing like that ever since. But that energy crisis, I thought, wow, maybe there's a different source of energy we can tap into. I mean, not only for gasoline, but for natural gas. And I remembered learning in a a science camp type of thing at Providence College. I learned that anaerobic bacteria, they generate methane. And I thought, geez, well, why can't we just harness that methane to supplement or replace natural gas to help address some of the energy crisis problems that were manifest at the time? And so what did I do? Well, where do you find anaerobic bacteria? Well, where you have decomposing organic matter. And I remember the fellow at Providence College said, the sewage treatment plant, there's a lot of decomposing organic matter there. So what did I do? I went to our local sewage treatment plant in North Attleboro, Massachusetts. That's my hometown. I went in and see the boss at the sewage treatment plant. And I said, I'd like some sludge. He thought I was crazy. Who goes to a sewage treatment plant and asks for a bucket of sludge? Because when the effluent from the town goes into the sewage treatment plant, there's a big container, if you will, that it passes through first. So all the solid matter, the particulates, settle to the bottom. And then the water continues on to go through a purification process. So I wanted some of that stuff at the bottom of the that first tank. And the guy thought I was crazy, but boy, we went to that tank and he took his little scoop, his really long scoop. He went right down to the bottom, scooped up some of that sludge. And I brought that sludge, well, first home, because this was after school. I remember my mom complaining about the smell in the garage, because that's where I kept it. And then the next day, I brought it into school, where I had set up basically a reactor vessel for the sludge and was able to collect the gas and measure the gas output per gram of sludge that I had in the reactor vessel. My calculation was that we could generate, in principle, half of the energy demand of the town, just based on the amount of sludge that went into that tank at the sewage treatment plant. So you just kind of follow your curiosity, and that's what I did as a high school student. You just don't care where you have to go to follow your curiosity. And if it takes you to the sewage treatment plant, then hey, have fun. You'll never forget the smell, though. I will never forget the smell. And being in Philadelphia here, sometimes sometimes there's sewer effluent gas coming through some of the grates in the city. So I never forgot that smell. But the key point is that you just follow your curiosity. And when you follow your curiosity, you're having fun. And that just continued on as I learned more and more about science and chemistry and biology, biochemistry. It just kind of broadens the field of curiosity that you can follow. So that's what I did. And that's what I would recommend that any scientifically inclined high school student or even younger do. Just kind of follow your curiosity about the world.
Yeah, that's so cool. And then what traits define a great researcher in their field, and what type of traits would you look for in a potential lab member?
A curiosity, a drive to discover new things. You have to work hard. So some evidence of being willing to work hard. Being able to troubleshoot problems is a useful skill. So I might ask someone, have you ever encountered an obstacle in a course, if not a research lab, and how did you overcome that obstacle? Looking for creativity, I suppose, because sometimes you really do have to think outside of the box in order to circumvent problems in the laboratory. So being knowledgeable about techniques and systems is important, but too much knowledge might be a dangerous thing because you might rule out the out-of-the-box experiments that might otherwise be successful and lead to new discoveries. So sure, you follow the standard procedures 95% of the time, but 5% of the time it's okay to explore things that you don't think will work because sometimes they might actually work.
And then just for our final question, what kind of like tips would you give to a high schooler who's looking to go into research in your field?
Work hard in your chemistry and biology courses because it's in chemistry where you learn about chemical bonds. It's in biology where you learn about proteins and enzymes. And so if you're interested in biochemistry, which has many names now, chemical biology is one such name. But if you're interested in science, in research at the interface between chemistry and biology, getting as much of a experience in chemistry and biology as possible is quite important to go into the field. And that's it should be easy to do even as a high school student. I mean, I had 2 years of chemistry in high school and 2 years of biology. So that second year, the course, once you get past the introductory stuff, I found to be quite illuminating and really kind of digging into things like protein structure, in the advanced biology course. And in the advanced chemistry course, it was a lab-based course all year. I mean, designing experiments, writing lab reports, learning about least squares fit of linear data and so forth. I mean, all of those are very valuable lessons that I draw on even to this day.
So like those are all the questions that I have for today, and thank you so much.